Okay, so we're going to practice, if you're quarantined, we're going to practice protein synthesis via these alien creatures called snorks. And first we see a table of genes that each of these creatures is going to have. They're all going to have all 10 genes shown, one for body covering, style, legs, and so on. And the thing is that each gene comes in two phenotypes. So you can have the hairless phenotype or the hairy phenotype for body covering, for example. So we're going to skip down to our first creature, and you only have to do one. You don't have to do two. So you can pick the first one or the second one. It's up to you. The first thing is we have our DNA sequence. Now, the nice thing about a codon chart, which I'll show you in a second, is that we have a shortcut with it. So we do not have to do the tRNA step. We just have to transcribe this stuff into mRNA. So all you got to do is you need to take the T and the A and the C and you transcribe it. So that would be what would pair with the T, A. Remember, RNA does not have a T in its vocabulary. It has a uracil instead. So this would be a U. And then it's just like pairing DNA together, except with RNA. Anytime you would use a T, you have to use a U. So we keep going. And we'll stop here. So we got, actually, I'll do the first line. So we got UAA. And then we got, we'll stop there. So here's the thing. So we've started transcribing it by just pairing up the mRNA bases to the DNA. And the nice thing is we're just going to take these codons and we're going to plug them into the codon chart. So our first one is AUG. So you can use either the top one or the bottom one. I'll show you how to use both. For the top one, the first base is in the center. So you could go A, then the next base is U, and then the third base is here. And so then you have methionine, which is met, and that's your first amino acid that you would string to it. Now, for the second chart, what you could do is it's more like a grid. So A means that you're going to have anything in this column, this row. The second base being U is going to narrow it down to this box because this is where the U column is. And then the third base is the one that is the deal clincher and tells you which one of those four options in the box it is. G is right here, so that means it's going to be met. That's where A, U, and G all intersect. So we just do that for each one. So this first one is translated to met. And then we do G, U, C. So we go G, U, C, val. AGC, AG, and C is ser. And then we got AAA, so that would be boom, boom, and Glenn. And then UAA would be 1, 2, 3, is stop. So what does that mean when it says stop? So you just write stop there, and a stop codon means that the gene sequence is over. So let's see what we got. We're looking for the gene for the body covering that matches this amino acid sequence. This is met, val, ser, glen. So we go up here to our chart, and met is the start codon. So that doesn't actually include the gene. It's just saying the gene is now starting, so what comes after it? So we have val, ser, lu or val, ser, lis. So let's see which one we got. Uh-oh. Looks like I made a mistake. AAA. I did. AAA is lis. Whoops. Happens to the best of us. All right, so that would be lis. Okay, so val, ser, lis is going to be hairy. And so that means that our first gene shows that this snork is hairy. So I'm going to do a little bracket here, And I don't need to include the start codon, but I include everything between the start and the stop codon, and this is Harry. Now, the thing is, you're going to see you just need to transcribe the whole thing of DNA. So do this process for all of it, but I'm going to give you a warning. You're going to have some DNA codons that don't code for anything. I'll explain that next week, but you might have some gaps between your start and your stops. So that's okay. Just keep transcribing the entire thing, and then when you have all the proteins coded for, then you can start to find where the genes are. So the thing is, like, step one, make sure you transcribe everything, and just do that for the whole DNA. And then once you have everything transcribed, this line, and so on, then you just do the protein synthesis part where you translate it into the amino acid codons. Then you can find the genes in it. So if you have any questions, send me an email, and I'll post a key for Monday.